This is part 2 of chapter 11. We have been talking about mitosis, and now I wanted to get into the specific phases of mitosis. So you've probably seen these before. You're going to want to know these for the chapter quiz. But mitosis gets divided into these subphases, and they are prophase, prometaphase, metaphase, anaphase, telophase. And I will go through a brief description of what's happening in each of those phases. And honestly, I'm not a big fan of this pro-metaphase, so I'm not super worried about that one, really. To me, it's all kind of prophase, but your book does make that distinction. Okay, so let's go through these one at a time. And what you're going to see in the next few slides is you'll have two images for each of the phases. So the pictures at the top are actually taken with fluorescent microscopes, where you have the spindle microtubules in green, right? So you can see all the spindle fibers, and then the DNA, the chromosomes, are labeled with red. And then at the bottom, you've got the cartoon version of what's going on there. So I'm going to just focus on the cartoons, but if you want to see the real pictures, you've got them there as well. Okay, so remember, interphase was, all the stages of the cell cycle that were not mitosis. And then we would begin mitosis by going into what's called prophase. And now in prophase, this is where that DNA is condensing, as we talked about in the last video. So it's getting wrapped around those histone proteins and condensed, right? Take it from the spaghetti to the raviolis, to go back to that dumb analogy. So we condense the DNA just to make it easier to move around. We see the spindle beginning to form, right? So the centrosomes are moving to opposite sides of the cell. The spindle microtubules starting to grow between them. And then in prometaphase, and as I said, I really just kind of still consider this prophase, but whatever, the nuclear envelope breaks down and the spindle attaches to the chromosomes. Okay, so if we're gonna segregate the chromosomes, we got to get rid of the nucleus because they're trapped inside the nucleus. And remember, the nuclear envelope is just made of membranes, so we basically dissolve away that membrane, and now the spindle has access to the chromosomes, and those spindle fibers attach right at these points called kinetochores. And yeah, I think I have already mentioned this, but yeah, kinetochores are those attachment points. And again, these microtubules are shrinking, they're growing, pushing and pulling the chromosomes around. So they're providing that force to actually drive mitosis. Metaphase is probably always the easiest of these substages to identify, because at this point the spindle has pushed the chromosomes into the middle of the cell. So they're basically just all lined up, sort of in a single file line in mitosis, just end to end to end. What's called the metaphase plate here in the middle of the cell. And now in anaphase, we are actually going to separate the two sister chromatids, right? So remember, each one of these chromosomes, because we'd gone through the process of DNA replication, is composed of two copies, right, attached to each other. There's two identical sister chromatids. So in anaphase, we split those apart. And as soon as they split, now they become full-fledged chromosomes. So we end up with this little temporary period where we actually have twice the number of chromosomes in this one cell. And interestingly, there's actually a sort of a protein glue that holds the two sister chromatids together. That protein is called cohesin, which is a good name for it. You might remember from the previous video, there's actually one of those cell cycle checkpoints right in the middle of mitosis, basically at anaphase. And what will happen is the cell has to stop at that checkpoint, but if everything looks good, like, the number of chromosomes seems to be correct, and there's not, weird DNA damage or something, then one of these cyclin CDK combos will show up and basically activate this enzyme called separase, and separase will essentially just chew up all the cohesin protein, right? Get rid of the glue that's binding them together, and allow those two copies, those two sister chromatids now to separate. Okay, so that was anaphase, and then in telophase, we're basically regrowing the nucleus now around each set. So as the microtubules here shrink, right, they're going to pull those daughter chromosomes to opposite sides of the cell. We will basically start to get rid of the spindle, regrow the nucleus around each set of chromosomes. That would be telophase. And I'm going to post up on Canvas just another short video that kind of goes through all these stages animated for you. Might make it a little bit easier to think about. And then lastly, we need cytokinesis to actually split the cytoplasm. And I just wanted to point out cytokinesis happens a little bit differently, whether you're talking about animal cells or plant cells. So in animal cells, you get formed what's called a little contractile ring. It's basically actin microfilaments and myosin motor proteins that work together to kind of just pinch the cell in half, right? You can see that happening here in this image taken with an electron microscope. So that ring just keeps getting smaller and smaller until the cell's pinched in two. In plant cells, that approach wouldn't really work because of the tough cell wall, right, that surrounds the plasma membrane. So you have to be able to pinch through the cell wall as well, and just not feasible. So what plants do instead is they just build a new cell wall. They call it the cell plate between the two nuclei. So they just start making a lot of cellulose fibers inside the cell and sending them in little vesicles to the center, to the cell plate, where they just accumulate and build a new wall. But the end result is the same, right? The cells then are physically separated from each other in cytokinesis. And I also wanted to remind you, just go back while we were talking about this whole process. We saw how important centrosomes were for growing the microtubules, but just to remind you that plant cells don't actually have centrosomes, but they do still grow a spindle just fine, so they're just growing it without the help of centrosomes. All right, so to end this video, I just wanted to point out how important the balance is, especially in a multicellular organism, between mitosis, right, production of new cells, and cell death, right, getting rid of old or unwanted cells. And I should point out there's two ways that cells can die. So there's necrosis and then there's apoptosis. Necrosis is the bad kind of cell death because it just happens due to tissue damage, right? And so when a cell dies by necrosis, basically the cell swells up and bursts, and so its contents now are, pushing on other cells, so you get swelling, you get inflammation, it hurts and it's uncomfortable. Whereas apoptosis is often called programmed cell death or cell suicide. And I use the analogy of it's like a mob hit where, the cell is just disappeared, right? There's going to be no trace of it. And apoptosis can happen if a cell is just not needed anymore. So a lot of times this is really important during embryonic development. You can think of it as kind of removing unneeded tissue. If you were given a lump of clay and asked to sculpt that into some particular shape, right? You usually do that by removing clay. So same kind of idea in an embryo, remove some cells by apoptosis. Or it can also be used to get rid of old or damaged cells. Maybe a cell has lots of mutations or DNA damage. You could intentionally get rid of it by apoptosis so that it's not causing any potential problems. And this is what apoptosis looks like. the cell will basically detach from any neighboring cells it might have been connected to. It will chop up its DNA into little pieces, and then the cell, literally, it's what's called blebbing. It starts basically just breaking itself down into these little pieces, and then those pieces get eaten by other phagocytic cells, and it is just gone, right? There's no trace of that cell after this process is done. So again, I'm mentioning this because there's this very fine balance, imagine in your adult body, between production of new cells by mitosis and getting rid of old cells by apoptosis. And so if we lose that balance, we can end up with tumors and maybe even with cancer. And so in cancer cells, these are cells that have basically accumulated lots of mutations over time, and they have lost control of that cell cycle, right? All those checkpoints, the cyclin CDK system is basically like a runaway train, right? The cell's not stopping at those red lights anymore. So we've lost that control, and those cells can move, right? We don't usually think about cells in our body moving around other than like circulating blood cells, but those cancer cells will take off and move to other parts of the body, metastasize. So again, this is really all due to typically mutations that have happened over the course of a lifetime. And anytime you have cells that are dividing too much, you end up with a mass of cells, and that's by definition a tumor. Tumors can be benign, meaning that they are still in the original location where they initiated. So typically benign tumors will grow more slowly, but there can be fast-growing benign tumors. And a benign tumor could still be dangerous, right, depending on its location and how easy or difficult it might be to be removed. But ideally, if you find a tumor that is benign, still in its original location, if you can remove it surgically, then it's gone, it's done, right? Whereas malignant tumors, by definition, a malignant tumor has spread from its original location. It's pushed into surrounding tissues, or worst case, it's metastasized, meaning cells have actually left that malignant tumor and may have moved through the bloodstream to distant parts of the body and established secondary tumors. So here's a cancer cell, and you can see because of these mutations, these genetic changes, this cancer cell, which is the yellow one in the middle of the image here, it doesn't even look like the other cells around it anymore, right? You can see it's totally different shape. This is a cell that's moving, so those DNA changes have really changed the appearance and the behavior of that cell over time. And just to tie this back in with our discussion of mitosis, most traditional cancer treatments, things like chemotherapy, they're basically just poisons. They're drugs that will just kill dividing cells. So, for example, classic chemotherapy drug is Taxol, and Taxol works by binding to microtubules. It just messes up the spindle, basically. So if a cell is going through mitosis, the spindle gets screwed up and it can't complete the process, which is fine. It's effective as a cancer treatment, but it's going to kill any cell that's dividing, right? Block any mitosis. So that's why you get some of these nasty side effects, like you lose your hair because you have dividing cells in your hair follicles. You get really nauseous because you have dividing cells that line your intestinal tract. So sort of the modern trend in anti-cancer drugs is to try to find things that are only going to target cancer cells specifically. So one great example of this is this Herceptin, which is an antibody that will just bind to a growth factor that's found in certain breast cancer cells typically. So it only binds to and affects those cells, and it's not going to bother the cells in your intestinal tract or in your hair follicles. So anyway, I will stop there, and we'll pick this up in the next section, talk about meiosis and sexual reproduction.